Hello, everyone. I'm Kara Strittmatter, and I'm the UMDF Meeting Event Director, and I will be joining the call tonight and helping Margaret.
And as we will be starting in just about a minute, Dr. Wallace, you can unmute yourself. Today we are very lucky to have Kendall Wallace Ph.D. He's a professor at the University of Minnesota Medical School at the Duluth campus. Dr. Wallace is a friend to the UMDF and has been a friend to the UMDF for a long time and is currently on the UMDF Scientific Medical Advisory Board. And we are so lucky to have him with us today. Dr. Wallace, will you tell us a little bit more about yourself? Oh, we've got you still muted. I'll unmute you. Dr. Wallace?
Yes.
Okay. Tell us a little bit about you.
Yes, well, thank you. So I'm a toxicologist by training and late career toxicologist. I built my career on studying the effects of various drugs and environmental chemicals that affect the mitochondrion and leading to a variety of different metabolic diseases. I discovered the UMDF when in the early years of the foundation, and I've become a very closely associated member, an enthusiastic member of the foundation. And I've had the privilege of learning a lot from my colleagues and the families there, and making a lot of friends, both professionally and personally within the foundation.
Thank you so much, Dr. Wallace. Little housekeeping before we get started. As you probably have noticed, you are all muted, and you'll be muted throughout the call. Dr. Wallace will be doing a presentation, and then we'll be accepting questions at the end. We will be communicating via the share button, which is, oh, not the share button, what am I saying? Through the chat button, and this is where you can post your questions. You put your question in, and type the question, hit enter, and the question will be there. When we are finished with the presentation, Kara and I will read your questions, and Dr. Wallace will answer them. So that's how that's going to work today. And Dr. Wallace, can you advance the slide? While we're waiting, before we get started, make sure that you have connected with the UMDF through our Connect box, which is connect@umdf.org, or you can reach out to me directly at margaret.moore@umdf.org. Mark your calendars for the symposium. It's in Phoenix this year. We'd love to have all of you there. And to learn more about ACT, our wonderful folks that had the idea to do this webinar and made it happen, you can reach out to us at umdf.org and we can get you connected. So the name of this webinar will be Mitochondrial Disorders of Non-Genetic Origin. Again, like I said, this webinar is being hosted by our ACT team, the Adult Advisory Council team. They think of ideas and come up with interesting educational opportunities for all of you. So without further ado, Dr. Wallace.
Well, thank you very much. And again, I feel quite privileged to have this opportunity to speak with you and share with all of you my perspective of mitochondrial disorders. It's important before we begin that you understand my perspective, where I'm coming from. As I said, I'm a trained toxicologist. I'm board certified in toxicology, member of the Academy of Toxicological Sciences, and past president of the Society of Toxicology. I am not a geneticist. I am not a physician. So I'm approaching this from looking through the lens of my being a professor of biochemistry and molecular biology at the University of Minnesota Medical School. So let me get to the next slide. These are the objectives, and basically I want to briefly walk you through how mitochondrial mutations, genetic changes in the mitochondrial DNA, lead to mitochondrial dysfunction and the multiple organ system failures. We'll talk about the metabolic processes that occur outside of outside of the genome of the mitochondria. the various pathways that result in mitochondrial dysfunction and how this affects the multiple target organ systems. I'll also want to pause and take a little bit of diversion and talk about hazard and risks, because many of the times we read in the media, both scientific and non-scientific media, about something being a mitochondrial poison or mitochondrial toxin. And in doing that, it's important to me that you understand the difference between something that tested as a positive hazard versus a real risk. So we'll spend just one slide on that. I then want to talk to you, after walking through the various metabolic pathways, I want to talk to you about the difficulty it is to distinguish, based on symptoms, blood chemistries, and muscle biopsies and such, the difference between a genetic and non-genetic disease, mitochondrial disease. And then I'll emphasize the importance of trying to understand what is the cause of this disease so that we can best design a therapeutic strategy that is most efficacious in treating that disease. The first few slides, many of you being engaged with the UMDF are going to be very familiar with this material, but I want to give a couple introductory slides so that everybody is kind of up to speed before we drill a little deeper. So most of you will recognize this on the left part of this panel, where the pointer should be, is a typical animation of a cell, and then it contains a number of mitochondria, tens to hundreds of mitochondria per cell, being an intracellular organelle. And then each mitochondria contains multiple copies of the genomic material, the DNA itself. And each of these mitochondrial genomes, they're a circular genetic code as opposed to the nuclear double helix. This is a circular code, and that DNA code codes for 13 proteins, as you can see around the outer rim of the genetic material, 13 different proteins, along with 21 transfer RNAs and 2 ribosomal RNAs. But these 13 proteins are all destined for the mitochondrial electron transport chain. That's kind of given into cartoon shape below the mitochondrial DNA, circular DNA there. I want to point out in this slide, here's your electron transport chain. Your complex 1, your NADH dehydrogenase, complex 2, complex 3 is your succinate dehydrogenase, complex 4, and then the ATP synthase is complex 5. This total complex it consists of more than 80 different protein subunits, those by the individual shapes that are included with each complex. I want to draw your attention to the bottom here. This is N stands for nuclear DNA. So these are proteins that are in these respective complexes that are encoded by the nuclear DNA. And then the bottom row here is the number of protein subunits that are encoded by the mitochondrial DNA itself. And you can see there's 7 of complex 1. Complex 2 is unique in that it has no mitochondrial encoded genes or proteins, and then 1, 3, and 5. If you do the math, you'll see this adds up to 13 protein subunits, as the previous slide said. So you can imagine if you have a mutation in any one of these genes encoding any one of these proteins, it's going to result in a dysfunctional complex. Each of these protein subunits are essential. They're vital to the overall function of the electron transport chain, which ultimately makes ATP. And again, the mutation and a defect in any one of these protein subunits will result in some degree of mitochondrial dysfunction. On top of that, I want to point out the fact that mitochondria are included in all tissue, all organ systems. So a defect in any mitochondria will affect a multitude of different organs, anything from cardiomyopathy to hepatotoxicity, diabetes, to vision disorders, to central nervous system disorders and such. So just want to emphasize the point that a mitochondrial disorder results in what we call a multiple organopathies or organ pathologies affecting. It's not a tissue specific type of disorder. Well, let me just back up. The degree or severity of the disorder of a mitochondrial disease is determined by the number of mitochondria within the individual tissues and how reliant that tissue, that function is on the integrity of mitochondria, their function. So we look along that same line. Now this is the same circular mitochondrial DNA, but now the proteins are on the inside of that circle, and on the outside of the circle, you probably all recognize this as the mitodisease map. And it lists the various types of diseases or mitochondrial disorders that are associated with mutations at each of these points along the sequence of the mitochondrial DNA. And just want to draw your attention to the multitude of different disorders, all of which trace back to a mutation in one of the 13 encoded proteins. Anything from MELAS to LANS to LS, ECM, NARP, myoglobinuria, we have encephalopathies, and just the multitude of different organ systems that will clinically will exhibit themselves or express themselves as a disorder. And so again, it's consistent with the fact that all organs, all tissues contain multiple copies of the mitochondrial DNA. So just to emphasize that point. This slide is where the UMDF, the foundation itself, is focused on genetic disorders or diseases associated with changes in the mitochondrial genome, defects, mutations in the mitochondrial genome. This is the UMDF slide. I come from it in a little bit different order, and this talk is about non-genetic mitochondrial disorders, disorders in the mitochondrial function that are not the result of mutations in this mitochondrial genome. Why is that important? This slide depicts it a little bit, is that depending upon the disease, there are varying degrees of risk that are based either on genetics or non-genetic risk factors. For example, type 1 diabetes, 88% of it is genes determined. Whereas melanoma, the genetic risk is only 21%. The majority of the risk for developing a melanoma is a non-genetic cause. And again, there will be a different pie chart, will vary depending upon which type of disease we're looking at. But these are just two examples. So I want to now move into some examples of non-genetic mitochondrial toxicities. We'll talk about different targets as well as different chemical sources for those toxicities. One of the primary ones that's talked about, it's closely related to the genetic gene-based disorders, are disorders in mitochondrial DNA replication. So here's your mitochondrial genome. DNA polymerase gamma or pol gamma is essential for the replication of mitochondrial DNA to make a second copy. And that's what is illustrated here. And a classic form of drug-induced mitochondrial disease or disorder is the reverse transcriptase inhibitors, they're anti-HIV drugs, or the fluoroquinolone antibiotics that interfere with the replication of the mitochondrial genome. Now, this mitochondrial genome needs to replicate because you have a dynamic turnover of the DNA within any given mitochondria, and so you need to keep replicating that genome. These drugs have been demonstrated to interfere with the mitochondrial DNA replication, resulting in what we call mitochondrial petite mutants. A closely related example is those of other antibiotics that here's your NARDs and the mitochondrial replication. But once those genes are synthesized, or genomes are synthesized, they then have to be first transcribed to RNA and then translated to the proteins that end up in the electron transport chain. So this is gene expression, transcription and translation. And these various antibiotics are examples of ones that have been reported to interfere with either the transcriptional or translational expression of these genomes. So it causes a type of injury that looks very much like what a mitochondrial DNA mutation looks like. So what happens when you can no longer synthesize these individual proteins, part of the complex? We go back to this slide and you affect one or more of these subunits, and in its absence, the mitochondria ATP synthesis, oxidative phosphorylation, is defective. and that cell becomes bioenergetically unstable, and you'll get associated types of functional deficits. So what are some examples? So that's gene-based, but there's also other examples of drugs or chemicals that affect these specific proteins, interact with these proteins and inhibit their activity. And the next slide are just examples of several drugs that affect complex 1, complex 2, complex 3, 4, and 5. Examples of drugs that have been reported to inhibit specifically these individual complexes within the respiratory chain. And you'll have access to the slide set so you can interrogate it further with more time after the seminar. There's also another way of interfering with this electron transport, and that is uncoupling it so the electrons no longer flow down the chain to be used energetically to phosphorylate ATP, but they're diverted across the to redox cycle, or they're pumped back into the mitochondria. These are called uncouplers, and there's several examples of drugs that have been shown to be uncouplers of mitochondrial oxidative phosphorylation, and they're listed in the blue panel here at the bottom. But these types of inhibitors are not restricted to pharmaceutical drugs. There's a variety of environmental compounds as well that have been shown to inhibit complex 1, 2, and 4, as shown here, or to uncouple, such as the wood preservatives, pentachlorophenol, some heavy metals, the pesticide, insecticide, herbicide, paraquat, and the insecticide perfluorosulfonamide are examples of environmental chemicals that also interfere with mitochondrial oxidative phosphorylation. And then there's these examples that of uncharacterized. There have been shown to interfere with mitochondrial oxidative phosphorylation, but it's not clear yet exactly how they work to do that. So they're provided to you just as examples. But I go back to this slide. It says, once again, if you inhibit these enzymes with a chemical, the result is not that much different than what you do if you mutate that individual protein subunit. On a functional scale, it's very difficult to discriminate whether it's a mutated protein or a protein that has one of these chemicals bound to it, rendering it non-functional. So what we talked about so far is the individual mitochondrial electron transport chain, but there's other ways of interfering with mitochondrial respiration as well, and that is to starve it of reducing substrates. So here's your electron transport chain. Complexes are shown here with the ATP synthesis. The citric acid cycle is also called the Krebs cycle or the tricarboxylic acid cycle, more descriptive of what really happens. And what it does is that it oxidizes metabolic intermediates within the mitochondrial matrix, and it generates these reducing equivalents, NADH or FADH2. And those are the molecules that carry electrons, the electron transport chain, that then power it up so it can make ATP. So this is a generator that generates reducing equivalents. If you were to inhibit the citric acid cycle, and I'll give you some examples, you would starve this mitochondria of substrates. So the engine would be sitting there idle, but the gas tank empty, and you would not be driving ATP synthesis, and the result would be not that much dissimilar than if you actually inhibited the protein itself, or you mutated the gene that encodes the protein. So here's the citric acid cycle that we just talked about. Here's the electron transport chain that we focus on for making ATP. And here's a couple examples of chemicals. These happen to be environmental chemicals that inhibit the conversion of the individual intermediates of the citric acid cycle to allow it to run. So they're putting the brake on the citric acid cycle, and you're no longer synthesizing the NADH and FADH2 needed to fuel the electron transport chain, the electron reducing substrates. So again, by inhibiting the citric acid cycle, you basically disengage the engine. But you can go further than that, because the citric acid cycle itself is powered by other substrates. Shown here are glucose and fatty acids. These are reducing substrates that when oxidized through glycolysis to make pyruvate, which then feeds from the cytosolic fluid into the mitochondria, where it then is oxidized, made into acetyl-CoA, which then feeds into the citric acid cycle to make NADH and FADH2. If you inhibit glycolysis, you indirectly starve or inhibit the citric acid cycle and cause a similar starvation of the electron transport chain. And a couple inhibitors are indicated in this slide. Likewise, fatty acids are oxidized within the mitochondria itself. They're transported from the cytoplasm of the cell actively into the internal parts of the mitochondria, where it is oxidized, fatty acids are oxidized, to then generate the intermediate acetyl-CoA that fuels the citric acid cycle. If you inhibit fatty acid oxidation with any of these example compounds, aspirin, ethanol, ibuprofen, tamoxifen, and the like, you will then starve the TCA cycle from acetyl-CoA, and it will slow down and not be able to fully energize the electron transport chain. So just examples of what we have with that. There's one more pathway that feeds into the TCA cycle that wasn't shown in this slide, and so I brought the next one in, and that's protein. Protein degradation occurs in the cytoplasm of the cell. The individual building blocks for the proteins are amino acids. They are transported into the mitochondria, which are then precursors to form glutamate, which feeds into the Krebs cycle, much like acetyl-CoA does, and then allows the Krebs cycle, or TCA cycle, to generate the reducing equivalents that drive the electron transport chain to make ATP. If you inhibit amino acid oxidation, you will, likewise, you will slow down the TCA cycle and starve the electron transport chain of these essential reducing equivalents, and thereby cause a bioenergetic deficit, ATP. Not much dissimilar than what you get if you inhibit fatty acid oxidation, glucose oxidation, or the individual electron transport proteins themselves. So I ask you, if we stop here and pause and let's just think, what would happen if you inhibit each of these? If you inhibit the TCA cycle, what would happen? Well, you would have buildup of all of these molecules that feed into it. Amino acids, the activated form of fatty acids, or your acylcarnitines, and the pyruvate that feeds in. Okay, that's typical of an inhibitor of a Krebs cycle, and it's typical of a mitochondrial disease as well, lower ADP or lower ATP concentrations. If you inhibited the oxidation of amino acids, they would accumulate within the cell. Likewise, inhibit fatty acid oxidation, you have accumulation of acylcarnitine and glucose as well. I share this with you because those are the bioindicators that are used clinically as a descriptive of a clinical descriptive of mitochondrial toxicity. Is if you take a blood sample and measure elevated levels of selected amino acids, of acylcarnitines, of pyruvate, lactate, and ATP, then that's all consistent with a diagnosis of a mitochondrial disease. And that's shown on the next slide. Again, the multiple organs, differential diagnosis, it involves multiple organs, and they're usually, these symptoms reflect a bioenergy deficit. And then either the blood or urine biochemistry will show the accumulation of lactic acid, a decrease in the pyruvate to the lactate concentration, which is indicative of inhibiting glucose oxidation. You'll have an accumulation of carboxylic acids, which is indicative of inhibiting the TCA cycle, and the accumulation of acylcarnitines and amino acids that respectively reflect the inhibition of fatty acid oxidation and protein oxidation. If you take a tissue biopsy, the ATP concentration will be low, the mitochondrial respiration will be inhibited, and you'll see a decreased activity of the individual complexes within the electron transport chain, complex 1, complex 3, complex 4, what have you. Those are all indicative of these types of diseases. Note that amongst these diseases, as they're also indicative of genetic mutation. So these types of biomarkers, if you will, are consistent with any of these types of forms of mitochondrial disease. The only way of getting definitive description of a genetic disorder is gene testing. Oh, excuse me, is genetic testing itself is the only way to to definitively discriminate whether it's a genetic disease or a non-genetic based metabolic disorder. So I want to go to the talking a little bit about hazard versus risk. So there's a word of caution. The long list of drugs that I included when we on the diagram or on the slide for mitochondrial complex 1 through 5 inhibitors, that's developed through screening assays for mitochondrial toxicity. And typically what you do there is you're looking, this measured response is inhibition of mitochondrial function of some form, and then the concentration or dose of that particular drug. And in the lab, we keep increasing the concentrations until we start to measure some degree of dysfunction in the mitochondrial function. Okay, that is what we call a hazard potential identification. We find that this concentration of this drug has a potential to interfere with mitochondrial function. Potential hazard. Does it actually do that? Well, that's dependent on risk. So here's your hazard potential dose response curve that I showed in the prior slide. That's your adverse effect. What's critical here is here's your therapeutic dose response. Hopefully it's to the left at lower concentrations or doses of the drug. And here's where you're getting actually the desired therapeutic effect, whatever that may be, whether it's improved cardiac function or a thyroid function or what have you. And you see that it too, it has the same shape as the hazard or adverse effects, but it lies to the left of it. So the real key here is what's called a therapeutic window. When are you able to see a therapeutic response without seeing an adverse response? And that's your therapeutic window. The risk of anything that is identified in a hazard screening bioassay of actually being a risk to the patient or to the individual will be determined by by the lack of overlap between the red and the black curve. This is the window of risk. So just because there's a published table of inhibitors, and I published these myself, just because a drug or a environmental chemical appears on that list of chemicals that have been shown to have the potential to interfere with mitochondrial function does not mean that you as an individual are at risk of having that adverse effect if you're being dosed down here on the red curve, for example. So please be aware of that as you read the scientific literature as well as the non-scientific literature. I just conclude from that, and this is across the board, not just mitochondrial poison toxicity, but toxicities in general, is the majority of hazards are not true risks, and thankfully so. So let me conclude here. That the points I want to make in this slide presentation, then we'll open it up for questions, is that there are numerous molecular targets for mitochondrial disease besides the genome. That would be like your tricarboxylic acid pathway, fatty acid oxidation, glucose oxidation, and the such. They share a very similar symptomology, which is a bioenergetic deficits in whichever tissues are affected. Again, they affect multiple organs. And they have these altered biochemical profiles. They have accumulation of acylcarnitines and lactic acid and amino acids, as well as dicarboxylic acids. They share that in common. A definitive differential diagnosis requires genetic testing to determine whether this is cause of a genetic disease, or one through your environmental exposures, lifestyles, occupation, and the like. It's important to distinguish the source because that will provide us the opportunity of more effectively treating the disorder. For example, if the source is a drug, we can discontinue taking the drug. If it's an environmental exposure, such as tobacco smoke, we can avoid secondhand smoke. So we have an opportunities to manage the exposure and therefore reduce the risk. We also have the opportunity is if it's a disorder is a chemical that interferes with fatty acid oxidation, then we could direct our therapy to that particular pathway without disturbing the pathways for glucose oxidation, amino acid oxidation, tricarboxylic acid oxidation, and such. so we can get more targeted, specific, and hopefully more efficacious therapeutic regimens. So it is important to distinguish the source of that disorder. So with that, I'll say thank you. I hope that this set of slides were informative, and I'll be glad to answer any questions that you may have.
This is Margaret. I'd like to go through some of the questions that we received prior to this event through a survey that was sent out along with the registration information. So here's one very specific question. Risk to mitochondrial damage. Inhalation of mothball fumes, naphthalene and paradichlorobenzene daily for several weeks annually or for 8 to 10 years. What do we know about those chemicals, and are they harmful to your mitochondria?
So I heard the word risk in that. And then I heard the question go on and kind of describe to me what their exposures are, 8 to 10 years and such. So the individual chemicals themselves, the naphthalene and such, we know that that's membrane reactive. We know that sufficiently high enough concentrations for a long enough period of time will disrupt the membranes in which the electron transport chain is embedded. We know that it will interfere with oxidative phosphorylation. The key is, has the exposure, the individual person's exposures, been high enough, been such that that did cause a clinically significant loss of mitochondrial function? So I'm not answering your question, but I am saying that the chemicals that were described, yes, they have the potential, the hazard, they're mitochondrial hazards, but the risk depends upon the degree of exposure.
Moving on to the next question I received. Drinking well water containing substantially elevated arsenic above the reduced federal standards permissible for public water sources for 2 to 3 years.
The question is whether that's a risk.
Is that a risk to your mitochondria? Is arsenic, is that the function with which it?
So yeah. So the answer is going to be kind of the same. Arsenic is known to, it has been shown to interfere with mitochondrial function. What we now have to do is ask about exposure. Has the exposure been high enough that it reached the levels, arsenic reached the levels in the body to actually affect that change, that adverse outcome? So it's the same answer as the first question. But let me add to this one, and that is that we as citizens, as individuals, we expect our regulatory agencies to protect us from these adverse effects, such as mitochondrial toxicity. And so we mandate the Food and Drug Administration, the Environmental Protection Agency, and others to set safe exposure limits. Okay, so in this case, the drinking water levels that are allowed in the drinking water, those levels are set on a precautionary basis. I'll remind you, EPA stands for Environmental Protection. And so when risk assessment and setting these standards, there's magnitudes of precaution that are taken to accommodate the unknown. And by that I mean that if we find that a concentration of arsenic in the drinking water for rats over 48 months is 10 micrograms per mil, then we will lower that. The agency will then apply safety factors to protect the vast majority of individuals. And they'll apply safety factors that say, well, a human is 10 times more sensitive than a rat. And so they'll take that number, and the published allowable drinking level will be magnitudes lower than what has been shown to be harmful to rats. So what I'm getting at here is that those levels, just because you're exposed over to levels above that, doesn't mean you're going to get sick. What it means is that you don't have a great deal of confidence that you won't get sick. I did not say that very well, but it's a precautionary thing. It's almost certain that if the levels are below that, you won't get sick. If the levels exceed that, well, we're not quite as certain, and we have to check it out, but it doesn't necessarily mean that you're going to get sick. I hope that I answered that okay.
It does. It's actually very clear, and I'm learning a lot on this webinar. I have one more from our SurveyMonkey survey, and then I have a couple from our Q&A. Somebody figured out how to use the Q&A, so I'll read those too. Botox injections over many years. What do we know about this? Do we know that this has any impact?
You know, I honestly, I personally do not know.
Okay.
I apologize.
Well, that was our last SurveyMonkey question, and I will move on to the ones in our Q&A, and then lastly, we'll move on to the chat feature, which we have lots of questions there.
Okay.
The first one that we received through our Q&A was, for non-genetic mitochondrial dysfunction due to an external factor, then can there be continual progression of mito symptoms if no longer exposed to the hazard?
The answer is yes. You know, the best example that I'm most familiar with is the anti-cancer drug adriamycin or doxorubicin. So yes, it's not the only example. There's others as well. And so these dysfunctions, these disorders far outlive the time, the residence time of that drug or that chemical in the body. So it'll be eliminated, and years later, you'll still have that deficit. With doxorubicin, you know, it's the clinical gold standard, is that a young person, a pediatric patient is treated for, let's say, leukemia with doxorubicin, effectively arrests that leukemia, treats it. The patient goes on, the individual goes on for many years, and let's say it's a woman. And then at the age of 50, either gets pregnant or has, let's say, a breast cancer tumor, is treated once again with doxorubicin, and her threshold is remarkably diminished. Threshold of tolerance for the drug, remarkably diminished. She's at a much greater risk of developing cardiac failure, which is the primary adverse effect of that drug. So yes, it far outlasts the lifetime of that chemical in the body. And my lab, as well as many others, are actively researching how that works, the mechanism by which that happens.
And the next question from our Q&A section, for those undergoing an operation with mitodysfunction, propofol is often used. What is the safe limit to be under with propofol without negative consequences to mitochondria? Are there exposure recommendations for common drugs published elsewhere, ibuprofen, antibiotics, etc.? Is there a limit? Is there a specific amount that works for everybody?
So yeah, I don't think there's one amount that works for everybody. There are ongoing recent studies, or basically there's been workshops that have been conducted trying to identify which chemicals should be avoided, especially in patients that have a genetic mitochondrial disease, primary mitochondrial disease. But that's all based on clinical experience as far as what they see as the outcome for the risk of that patient. I don't know personally what those thresholds are that this questioner asked, that you need to stay below in order to be safe, or whether you should avoid it entirely. So I can't answer that question as a non-clinician.
All right, the next one. Can a person experience symptoms of mitochondrial dysfunction years after long-term exposure? An example would be exposure to trichloroethylene, sorry, I'm not good at these words, via vapor direct contact by swimming or playing in a creek water during childhood, along with a long-term vapor intrusion exposure throughout life.
Well, so hypothetically, it's possible. That's my opinion. I only base that on the fact that one of the prior questions is that if you cause a mitochondrial injury, can it outlast the duration of the exposure and go, months or years beyond? And I answered that in the context of our experience with doxorubicin. So with trichloroethylene, I have to say if it works for doxorubicin, then if I had to guess, I would guess that it's plausible. But the chance of it happening, I'm not certain of that. We'd have to undergo further testing. And those tests are available. The differential diagnosis mentions several of the examinations or procedures that could be performed in order to see if there is a mitochondrial dysfunction, that long after a chronic exposure. What we have to understand, and if I may kind of divert a bit, but the body, the cells, individual cells of the body are engineered in such a elegant way, in that the last thing that cell wants to do is to die. And so in a response to an exposure such as this, with mitochondrial stress, if you will, that cell then triggers events within the cell that helps to boost its bioenergetic capacity. It makes more mitochondria. And it tries to then dispose of those mitochondria that are dysfunctional. And then it shifts its metabolic needs from mitochondria to non-mitochondrial sources, such as glucose oxidation, glycolysis. And so that's how we withstand a lot of these pressures, whether they're chemical exposures or emotional or physical exercise or whatever, is that the cells individually are equipped to compensate and adjust to survive in this different environment. So that's a highly protective, life-sustaining type of compensatory feature that most all cells have.
Thank you. The next question, why is valproic acid so toxic to mitochondria for people with POLG mutations?
Well, let's see if I can go back to the slide that I want on here. That you basically double hitting the mitochondria. How do I want to do this? You're hitting them with two different, kind of left-right punches, and that. First of all, you can see this slide, I hope. First of all, if you look at the electron transport chain with POLG, you have fewer of these because you have a mutation, or you're not allowing the mitochondrial genome to replicate, so you have fewer mitochondrial genomes, and so fewer genes encoding for these proteins, so you have fewer electron transport chains. Then on top of that, you're going to, I picked the wrong slide, you're going to, there it is, you're going to inhibit fatty acid oxidation. So you basically hitting the mitochondria in two different dimensions, both of which result in inhibition of ATP synthesis. So that's why a person that has a POLG deficit or mutation is more sensitive, should be more, in my opinion, more sensitive to any of these other types of inhibitors of the substrates themselves, glucose oxidation, fatty acid oxidation, and the likes.
Okay, for those, here's the next one. For those that have had genetic testing, muscle biopsy, and all the standard mito blood tests are negative, but a person has strong mito symptoms, are there tests often not used that can look for dysfunction that most doctors wouldn't know about? So are there some laboratory kind of things that could be done to find out more about whether they've been exposed to toxins?
So I'm assuming that this person, they talk about all the tests that have been done, it includes everything here, perhaps even genetic testing. And then the question is are there other tests that can be performed? And the answer is yes. What is the return on investment in doing that? given enough time and enough money, we can probably find something wrong falls outside the range of normal. Whether it's meaningful from a clinical standpoint is questionable.
And the last one from our Q&A is, well, there's actually two. One is, if my sister had DNA testing indicating short-chain acetyl deficiency syndrome, but not diagnosed because she didn't follow up with a geneticist, and she's being treated for MS, is there an overlap that you know of?
I'm not sure I understand that question.
Well, I'm guessing that there was some DNA indication of a mitochondrial disease, but somehow the diagnosis has ended up as MS. And are these things commonly seen together, and do these same kind of toxicity issues apply?
Well, so I don't feel comfortable answering the first part of that question, because that's a clinical question. Do these disorders, are they comorbidities? Do they associate with each other? I'm not sure of the clinical history with that, so I can't answer that question.
Okay. And then the last one is, can you please repeat the name of the cancer treatment drug?
Oh, sure. So the trade name is adriamycin, A-D-R-I-A, mycin, M-Y-C-I-N. And the generic name is Doxo, D-O-X-O, Rubicin, R-U-B-I-C-I-N. That is one of the most highly prescribed anti-neoplastic, anti-cancer drugs in the U.S. because it's a broad spectrum of efficacy and its potency.
Wonderful. Kara, are you looking at the chat feature, and can you get started on those?
Sure. The first one is, what would a mito reaction to lidocaine look like? And ibuprofen.
So lidocaine would basically cause just a general slowing down of oxidative phosphorylation. Lidocaine basically gets within these membranes and dissolves the membranes, if you will, and these membranes are necessary to stabilize the individual respiratory complexes. And so it'll be very difficult. Right now, it's essential. A healthy intermitochondrial membrane is able to effectively partition these protons that are hydrogen ions that are pumped out from diffusing back in. But as you start putting this membrane to sleep with lidocaine, these protons will start leaking back in. And so it'll interfere with oxidative phosphorylation and ATP synthesis that way.
Okay. And then she also asked about ibuprofen, but I think you might have, did you cover that with the poll, G?
I think so.
Okay. Is there mitochondrial DNA mutations? If there are mitochondrial DNA mutations, how often are these mitochondria recycled through mitophagy?
Yes. So mitophagy, I'm glad that this questioner is familiar with that. So in cardiac muscle, it's estimated that the average cardiac mitochondria is recycled, if you will, every, I believe it's, is 2 days or 2 weeks. But that gives you kind of a scale of what it is. It's not on the hour, it's not several months. It's within a matter of 2 to 14 days. So was that all the question there?
Okay.
How often did you cycle?
Yes.
Yeah.
If you have mitogenetic mutations on recessive genes, is your risk of harm from environmental factors increased?
I would have to defer that question to a geneticist, a mitochondrial.
Okay. And I think we've already asked about PROIC. Someone is asking, has the mitotoxicity list been updated with these drugs? And I can answer that somewhat. There is a paper that will be posted. I think we'll be sharing it very soon probably through UMDF news, and it'll probably be posted on our website. But there is a paper from a group of doctors from the Mito Patients, the International Mito Patients Organization, is going to be posting it as well. Matter of fact, I think they already have it on their page there. So we can share that link after this call.
Yes, actually, we have it on our website, Kara.
Oh, is it already posted? Okay.
It just went up. It's under Find Support, and it's in the Mito 101 section of the Find Support tab. So if you go to www.umdf.org, go to Find Support, and to the and then you'll see the Mito 101, and the article is right up at the top with the new list of.
Right. And it's interesting because this literally just came out, and after hearing your talk, Dr. Wallace, I'm seeing a lot of the same things I saw in the paper. So it's interesting.
Can I just expand on that? The important thing that I want to say is that the list that I have here, these are drugs that have been showed through screening, drugs and chemicals, through screening tests for mitochondrial hazard. They have the potential, but the publication, this group, the patients groups, the International Mitochondrial Patients Group associated with UMDF, they're basing their recommendations on actual clinical experience. So they're looking at risk,
right.
And so it's important for the listeners to appreciate that difference. Their list is going to be a lot smaller than a list that's developed through mitochondrial screening. So I welcome that publication.
Thank you. Are mitochondria replicating when cells are not dividing?
The answer is yes, because there are several tissues where cells don't replicate. Nerve cells, cardiac cells, they're mitotically fixed. And so when those cells die, they're not replaced, unlike several other tissues. So yes, and within, as I mentioned, within those cells, mitochondria turn over on average every 2 to 14 days.
Okay. Would it be possible for quarrying near your home to have consequences that may be toxic to people? For example, a slate quarry, especially young children.
So the operative word there is possible, and the answer is yes, it is possible, because a lot of the key elements of the Earth's crust are like heavy metals, it's been demonstrated that heavy metals at sufficient concentrations to interfere with mitochondrial function. So yes, it's possible. Is it probable? Well, that kind of depends upon exposure. Again, I don't mean to be evading,
right,
questions, but the fact is we need to do an exposure-based risk assessment to answer these questions.
Okay. And I don't know if this is too clinical or not. What routine blood tests should a child have who has mitochondrial disease of non-genetic origin? And should they have tests done when they're well or sick when experiencing a mitochondrial crisis?
That you're right. That it should be. I'm going to defer that to a pediatric clinician. Yes.
Okay. All
because
right.
These individual diagnostics that I had here, they differ with age, and so the concentration of acetylcarnitines, for example, in the blood differ for a child than they do in a young adult or in a geriatric patient. So a pediatric biochemical, a pediatric neurologist or physician would be the appropriate person to ask.
Okay. How would one go about determining if a medication overdose caused mitochondrial myopathy? And if the damage to the mitochondria is significant and debilitating, have you ever seen the damage reversed? That's part of it. Question.
Okay, let's read that again. So
how would one go about determining if a medication overdose caused mitochondrial myopathy, and if the damage to the mitochondria is significant and debilitating, have you ever seen the damage reversed?
Okay.
This may be too clinical too. I'm not sure.
So this is a case where you're seeing the questioner specifies a myopathy. So an individual experiences myopathy. They associate that with the consumption of or administration of a high dose of a medicine, of a drug. How do you determine whether the myopathy was caused by that administration? So you'd have to rule out all other possible associations, would be one. That's just by temporal association. You have to rule out all other associations, and you'd have to look at the plausibility. Is that drug, what does it look like as far as a therapeutic window for adverse effects? And did you exceed that, at your plasma level or tissue level, exceed what this window is where risk becomes, where the hazard is actually realized? And then will it be reversed? I don't know for sure because we're talking about a lot of hypotheticals here, but my guess would be yes, at least in part, it would be reversed. Perhaps not fully. I don't, but too many hypotheticals.
Sounds like it was a child. It says our daughter was given a maximum adult dose of a medication and went from healthy to no longer being able to walk, talk, had significant lung damage, etc., and has been diagnosed with non-genetic myotomyopathy.
Tragic.
Yeah.
Let me say that first.
Hard to tell.
Yeah.
Yeah. Are you aware of Suboxone, Suboxone, used long-term being harmful to mitochondria? The ingredients are porphynorphine and I can't pronounce all these. BUPRPORNORPHINEE /NALOXONE.
Okay, now the question about those is, am I aware of?
They've used long-term being harmful to mitochondria.
Yeah, I'm not familiar with that literature on this drug combination, to be honest with you. So.
Okay. Okay. What effects do pathogens such as EBV and Borrelia have on mitochondria?
You're kind of getting out of my wheelhouse here.
Okay. Okay.
I'm a chemical toxicologist. I know there are interactions.
Yeah. All right. That's all I have because the rest are repeating, I believe.
I actually have one more that popped up in the Q&A.
Sure.
Oh, okay.
It says, is a large deletion a sign of a non-genetic mitochondrial disease? If you see a large deletion, is that what you would, potential thing that you'd expect?
I'm aware of examples where the large deletion is a result of a non-genetic stress or source, environmental or otherwise type of exposure that cause large deletions. So it's consistent with a non-genetic disorder, but that's not to say there's not examples of gene-based large deletions as well.
I think that might be it. Oh, wait, another one's popping up. Okay, here's an interesting question that I never thought of. Is there a list of chemicals that were found to not impact your mitochondria?
So you can never prove no. Is that, and I'm being honest, not facetious, is that you can never disprove the null hypothesis, and that somebody could derive some sort of test that might show an effect. It may not be any of the the conventional diagnostics that we use, but if you do enough tests and you test enough different things, that you may come up with a positive result, a positive or an adverse effect. In fact, a statistician will tell you that the probability is that it would support that. In other words, in my lab, we use, for statistical purposes, we say the chance of a probability of less than 5%. Would that mean that just statistically, 1 in every 20 experiments that we do will give us a false answer? So yes, you can never prove anything that is absolutely not a mitochondrial toxin. And let me expand on this, is that it takes a lot of energy to hold that mitochondria together. In order for the mitochondria to make ATP, it has to maintain all of its membranes. It has to keep this intermitochondrial membrane intact, highly functional. That's not it. Here it is. These membranes have to stay intact, and it has to hold that respiratory chain intact and in position so it can pass electrons onto its nearest neighbor and do its job. And it takes energy to maintain that, replace these proteins periodically, make new mitochondria, dispose of old ones. That's all ATP generating, ATP requiring energy that it needs. If you start giving enough of a chemical, you will start affecting the health of that cell, and it will begin the dying process. And in the dying process, it runs out of it, depletes itself of ATP, what it needs to maintain this mitochondrion. And so with time in a dead and dying cell, this mitochondrion will start falling apart. It will start not using oxygen, making reactive oxygen species, not making ATP, and it will look very much like a mitochondrial poison, when indeed all of that loss of function is secondary to the fact that you're punching holes in the cell membrane, or the cell's dying for some other means. So you can't show that. So there's a lot of the literature. I found examples in the literature where in my opinion, they erroneously label some chemical as a mitochondrial poison, when indeed you look at their data and they're looking at half dead cells. Half the cells they're looking at are dead. And so I wouldn't expect those mitochondria to be functional. I'll get off my podium on that. It's something that bothers me as a researcher.
We have one more chat, but I'm not sure that this one is too clinical, about nerve and muscle damage if it were to occur, I guess, from some exposure. Would it can get worse just because of age? I guess the real question is, does age have a role in any of this?
Well, let me talk in the theoretical, and the answer is yes. As we age, and I'm living proof of it, as we age, our reserves, our mitochondrial capacity decreases in most tissues, most cells. The mitochondrial capacity, that's maximum capacity, exercise capacity, it declines as our cells and tissues age. And when I was younger, I had a reserve capacity that exceeded what I really needed to use. So I was only using, let's say, 60% of my mitochondria in my heart. But now that capacity has declined as I age, and now it's only maybe 10%. And so now I can't exercise. My exercise capacity, maximum capacity, deteriorates. So yes. So in the background of all these chemical things that we're doing to, we're exposing our mitochondria to, in the background, with age, our mitochondrial capacity is declining. So we no longer have those reserves. And with that, we become more vulnerable, more susceptible to those chemical exposures.
Okay, and now I'm going to ask a question out of the Q&A. Could a mother's exposure to chemicals when in utero cause her future child to have a genetic mitochondrial disease? Or non-genetic, I guess.
I was already to answer that until you used the word genetic. So for a non-genetic, so that'd be drug or environmental caused disorder, the answer is yes. These chemicals they all have differing degrees of ability to cross the placenta. And in fact, the same is true for nursing infants into the breast milk. And so chemicals, depending upon their physical chemical properties, how water soluble or lack of water soluble they are, will cross the placenta, and they will be then distributed in through the cord blood and in the media that's bathing the infant, the fetus, developing fetus. And so the answer is yes, and there are several examples, experimental examples where this has happened. By that I mean, rodents and rabbits and mammals experimentally. So there's examples that has happened. There was one other thing I was going to add to that. Oh, and then what makes it worse, and with the slide that I have up as appropriate, is that during development in utero, these individual organs they have spurts of windows of development during that in humans, that 9 months, where they're more sensitive than on either side of that window, either before or after. But there's windows of susceptibility during the development of that fetus within the womb. And if that chemical, drug or otherwise, crosses the placenta and it exposes the fetus during that critical window, they have a much more higher probability of contracting whatever that mitochondrial disorder is than if that exposure occurred outside that window of time.
Thank you. For a person with mitochondrial dysfunction, does overdoing it cause mitochondria to die? That's not really non-genetic, but
boy,
that's getting into maybe more clinical.
Well, I think I understand the gist of that question, and I talked about how the cell doesn't want to die, and it does all sorts of things in order to compensate so it can survive in a changing environment. Exercise is one of those changing environments. And it happens to whether you have a mitochondrial disease or you don't have a genetic disorder, or you don't have a mitochondrial genetic disorder, is that by exercising, you're creating a condition where your cells are saying, I need to compensate, I need to strengthen my ability to make ATP in this new environment. And so I'll do that by expressing the mitochondrial genes I have now and making more proteins for more mitochondria. Plus, I will actually increase the rate of replication of my mitochondria, both the genome as well as the whole mitochondria. So it's really, it's like becoming more fit. You think of the mitochondria as a muscle itself, is that the more you use it, the stronger and more fit it gets.
Thank you. And I do believe, Margaret, I don't see any more questions in the chat.
I think that's it. Oh, actually, someone just popped up in the Q&A.
We'll take one more and then we'll
Interesting one. For non-genetic mito, do we not call it mito disease?
Oh, I'm so glad you asked that question. My good friend, Dr. Bruce Cohn, and I go back and forth on this because, Dr. Cohn is a pediatric neurologist. He really knows his genetic mitochondrial disorders, and he's one of the pillars of the UMDF. In fact, he chairs the medical advisory board, I believe. It wasn't that, I think it was the last meeting of the United Mitochondrial Disease Foundation. We're sitting together, and he called a genetic-based mitochondrial disorder primary, which then implies that my kind of non-genetic mitochondrial disorder must be secondary. And so I ribbed him about that, and we kind of laughed back and forth. But in the literature, they're tending to refer to a genetic disorder as a primary mitochondrial disease, and a non-genetic, the stuff that I talk about, as not primary, but perhaps secondary. But it's just mnemonics. As long as we know what we're talking about, whether it's genetic or non-genetic, that's, I think, the important part as far as the well-being of the patient.
Well, I think that's it. And, thank you so much, Dr. Wallace, for being with us today. I do want to do a little bit of housekeeping with our folks on the line. We will be sending you this recording so you can review these slides again. And if there's anything that you missed, you can listen to it again. Might take me a day or two to get that out to you, but look for that in your email in the next bit. I will also send, along with that, the new Mito medicines to avoid list, so you have that.
Right. Some of the attendees are asking for a copy of the presentation. Dr. Wallace, did you want to provide it in a PDF maybe, if you're okay with that? we can talk offline.
The slide deck and PDF, I'll send that to you yet tonight or tomorrow morning.
Okay.
Perfect.
Very good.
Great.
All right. Thank you all.
Yes, this was wonderful. And thanks everybody for being here. And thank you.
Great questions.
Yes, really good questions. And I think all of us learned something. So Dr. Wallace, thank you so much. We really appreciate your expertise in this topic.
Well, thank you. I appreciate this opportunity, and I too appreciate all the good questions that came from the listeners. I wish you all well.
Thank you.